As a child, I studied art, and I've learned that by drawing lines on a canvas, we cut the patterns we see all around us in the nature. In my youth, I became interested by science, and I've learned that patterns contain and underlie mathematical structures, but in a coded form. By deciphering such codes, scientists invent theories predicting regularities observed in the world. Working on my PhD on ephemeral rivers, I became passionate about patterns formed by flows interacting with sand, stones, and plants. I call such patterns islands in the streams. Patterns formation is an orderly outcome of self-organization processes, where some islands of predictability arise in a sea of chaos inside initially disordered system. Among myriads of complex systems on Earth, rivers represent a most vivid example of self-organizing system, which involves interactions between four main elements: litho, hydro, atmo, and biospheres. Fluvial systems are immense and difficult to study in nature. A common scientific practice is to examine the components of such systems separately in simplified laboratory experiments. Working in labs, I've learned that simple experiments increase precision, but their results do not necessarily reproduce reality. The reason is
half a bee, philosophically, must ipso facto half not be. Do you see?
For myself, I discovered a way to solve this philosophical dilemma when, as a postdoc, I joined the IGB scientists in their development of the River Lab. This approach offers a methodological bridge to connect field and laboratory methods and a platform for a cross-disciplinary dialogue. Early River Lab experiments were focusing on the interactions of river flow and biota. In this film, I present my vision about expanding the methodology of the River Lab to a wider scale of fluvial processes acting within the river channel corridor, with a tighter link to the biodiversity problems. When most of us think about a river, we imagine a pastoral picture of water smoothly flowing across green meadows. The scientist's view of a river is a water body within a corridor of adjacent floodplain, a lower area inundated during seasonal floods. A flood happens when rains or snow melt cause the river to rise. Many people think that floods are bad, but in fact, floods are essential to the health of river ecosystems. Natural rivers flood almost every year, and by inundating the floodplains, form wetlands, which provide critical habitat for aquatic and terrestrial organisms. By moving gravel, wood, and pollutants in huge amounts, the flooding river strongly affects populations and communities of organisms. In ecology, an event that drastically reduces the size of population is called a bottleneck. Thereafter, a population with a smaller genetic diversity remains to pass on genes to future generations. In a broader sense, any flood is a bottleneck event, which is often highly chaotic. But morphology of a river provides a mechanism of self-organization into this complex system. I'm going to illustrate it with a very simple experiment. The first bottle empties longer because of the highly turbulent character of fluid motion at the bottleneck. In the second one, which was pre-rotated, the vortex introduces a certain order that makes the fluid motion smoother and faster. Natural rivers with their floodplains work much like the first bottle, while in drained rivers the floods pass through their canalized channels similarly to the second one. In fluvial environments, the retentivity and dispersity of transported matter are essential for ecosystem sustainability and biodiversity. In the planned research project, named Self-Organization Mechanisms of Intersphere Interactions as the Drivers of Biodiversity in Dynamic Fluvial Ecosystems, I will study such complex mechanisms using as a proxies to interrelated components from both inorganic and organic realms in water, air, and sediments. At the core of self-organization processes are interactions between organisms and their environment through scale-dependent feedbacks and behavior-driven phase separation mechanisms, which have been illustrated as important across different organism scales, ranging from molecules to ecosystems. Scale-dependent feedbacks play the key role in conversion of initially uniform pioneering vegetation on flat gravel bars into forested floodplains on complex, rich slow terrains. Dispersal of mobile organisms is the major factor of behavioral-driven phase separation, which often depends on the local densities of organisms related to the availability of habitats. As a model ecosystem, I choose the Tagliamento River in northeast Italy, which is the last intact ecosystem of European importance. During my PhD and postdoc trainings, I have spent in total about 1 year working in the field on this river. Ecological studies carried out on the Tagliamento during the past 2 decades provide precious information for my research planning. The Tagliamento floods every year in winter. When flood ends, it leaves bars of bare gravel and sand. Soon, they are colonized by vegetation. Colonization occurs in 2 modes. Previous research focused on propagals, such as driftwood anchored during floods. In this mode, individual seedlings grow fast, but the area colonized by plants increases relatively slowly. In my project, I will focus on a less studied fast colonization by seed rains. In spring and early summer, riparian trees emit seeds dispersed by winds and randomly covering gravel bars. Additionally, the seeds that fall on moving water settle along the water edges of bars. On the Tagliamento, diurnal winds control the seed rains. In the mornings, colder air from Alps descends towards the Adriatic coast. In the afternoons, warm air moves upstream. When the surface of unvegetated gravel bars can be heated by 20 degrees higher than ambient riparian woods. Such temperature gradient induces thermal convection and lateral secondary circulation of the air. These secondary circulations control effectiveness of the seed rains.
Not be exaggeration.
With my mind.
Some time ago, we sampled insects and wind profile across the neutral boundary layer. This preliminary study focuses on the effect of vertical thermal convection induced by the effect of temperature heterogeneity across the floodplain. We measure vertical profiles of winds and sample flying insects and seeds using 20-meter tall portable towers with an ultrasonic velocity meter and a string of sticky traps. By sampling at different locations, we obtain patterns in the local airscape structure, which controls the dispersal of seeds and insects. The effectiveness of seed rains can be estimated as the proportion of emitted seeds to the number of recruited plants. To estimate this, I will perform seed trapping and germination experiments. When seeds germinate, they form randomly distributed meadows. Recurring floods trigger the scale-dependent feedbacks. In streamwise direction, closely located plants shelter each other and reduce hydraulic stress. But in the lateral direction, the jetting of flow around groups of plants increases physical forcing and erosion, which may uproot the neighboring plants. Stagnated water in shallow wakes behind plants promotes deposition of fine sediments and nutrients. This facilitates predominantly longitudinal dispersion of vegetation. The strength of morphodynamical processes increases with the development of riparian vegetation. Scouring and deposition on a scale of a floodplain vegetation mosaic produces a distinct topography composed of depositional ridges and erosional canals called the parafluvial ponds. These ponds are the natural habitats for the myriads of aquatic insects. Let's make a short tour into this fascinating world. Parafluvial ponds on the Taniamento floodplains are the home of inland floodwater mosquitoes, which hatch when ponds are inundated. In few weeks, the larvae become insects feeding on nectar, but female mosquitoes need a blood meal for reproduction. The mechanism of behavior-driven phase separation in mosquitoes and deer flies is driven by host-seeking behavior. In my study, I will focus on roe deer as a major host for both aquatic and terrestrial blood-dependent insects. Deer use regular trails in their diurnal daily transitions between feeding and resting grounds. Their breath emits odor in a plume, which direction and spreading rates are controlled by winds. Odor plumes are main drivers of hemotaxis in mosquitoes. In the near field, such plumes are similar to wakes, forming past vegetation patches. We will study movable plumes in floodplain vegetation mosaics at different stages of development and under different airscape conditions. We will apply visualization techniques, winds profiling, and insect trapping. To relate the behavioral-driven phase separation mechanisms to dynamics of floodplain vegetation and morphodynamics of terrain, we need to study such mechanisms together with scale-dependent feedbacks. At a scale of a river ridge, we will carry out field monitoring. However, detailed insight into feedbacks will be obtained in controlled field-scale experiments. Previous hydrodynamic research in riparian vegetation focused mainly on patches on a flat riverbed. This summer, we have carried out a preliminary experiment to explore three-dimensional effects imposed on a shallow wake by a droplet-shaped bedform with the model patch. These bedforms are intermediate in a build-up of rich slow terrains. I plan to extend river lap methods to study the role of scale-dependent feedbacks in formation of complex flatland morphology. I will use the river lab infrastructure, an instantial of loam, model vegetation, acoustic Doppler velocimetry, remote sensing, and visualization techniques. The results of experiments and field measurements will be used to develop analytical models, which will be further applied to parameterize, extend, and validate numerical models of self-organization processes. By running numerical experiments with different scenarios, we will explore the effect that dynamics of habitats driven by intersphere interactions, together with behavior within the community, cause on the dynamics of mosquito populations and their dispersal. In such exploratory numerical studies, I will carry out analysis of ecosystem stability to understand how small perturbations in factors driving self-organization processes in flatland ecosystems will affect recovery of their biodiversity after major disturbances. This is a fundamental question for the sustainable management of river floodplains under the threat of ongoing climatic changes. In essence, the Zorg4Bio project will search for clear scientific explanations of a butterfly effect in dynamic fluvial ecosystems. First proposed in A Sound of Thunder by Red Bradbury in 1952, this fantastic idea is still inspiring, with the core message: small things matter the most.